Today I want to show you how to build a DNA molecule. So remember, the monomer for a DNA molecule is going to be a nucleotide. There are three main parts to our nucleotide. So with our nucleotide, we're going to have our monosaccharide, the sugar, and in this case, because we're dealing with DNA, that sugar is going to be deoxyribose. Now the important thing about our sugar is that on our sugar, implied at each of these nodes is going to be a carbon atom. And those carbon atoms are going to determine the directionality of this sugar and eventually the directionality of our entire nucleotide monomer. So this sugar over here is given the position or the labeling of the 1' sugar, the 2' sugar. Here's our 3' sugar, our 4' sugar, and what's interesting about our 5' sugar is that it's outside of this main kind of ring. Now, we don't really care about the 1, 2, and 4 carbons in our sugar. We really care about the 5 and the 3'. So in all of my future nucleotides that I'm going to be drawing here, when I'm building my one strand, my one polymer of DNA, I'll only write the 5 and the 3. Coming off here from the 1' sugar, we're going to have our nitrogenous base. And over here attached to our 5' sugar, this is where we're going to have our phosphate functional group. So this is our one nucleotide monomer, a sugar, nitrogenous base, and phosphate functional group. Now in order to make one strand, one polymer of DNA, we are going to be bringing in each nucleotide monomer in this direction. Our nucleotide monomer is asymmetrical. Because of that asymmetry caused by our carbon atoms, the directionality of our sugar, our DNA polymer can only be built in one direction. It can only be made from the 5' sugar, carbon on the sugar to the 3' sugar. So the only way that my next nucleotide can come in is down here below my first one. For the sake of this demonstration, I'm going to abbreviate nitrogenous base with N base, and over here I'm going to abbreviate phosphate functional group with P group. Here is my 5' sugar, and here's my 3' sugar. My next nucleotide monomer can only come in down below, going in this direction. And again, 5 and 3 carbons on the sugar. Now in your own notebook, I hope that you are drawing this picture along with me. Again, that physicality of drawing this seems to help people really understand how this one strand, this one polymer of DNA gets linked and bonded together. If we were in class, I know that students in their notebooks would be drawing this as I was drawing this on a board. So hopefully you are drawing this at home. Just for continuity, I am going to draw this final nucleotide monomer down here, though it gets cut off a little bit, just so we can understand that this would keep going, that continuity in our modeling. So this DNA molecule is being built from the 5' sugar to the 3' sugar. This also gets the term our leading strand. It's easier to make, and just so you know, the textbook and a lot of the videos that you're going to see use this term leading strand. I don't really care. It's not something that I require, not something that I use in this class at this level. So this is just one strand of DNA. Our DNA molecule is going to be two strands, two polymers, that have come together to make a DNA molecule. In order to make the second strand, my nucleotide monomers are still going to fit together in the same way. They're going to fit together from the 5' sugar to the 3' carbon on the sugar. The only difference is because I have to make and build the second strand in the opposite direction. So I have to start down here and do some Cirque du Soleil gymnastics in order to make my sugar. This is my 1' sugar carbon, my 2'. This is my 3' carbon. Here's my 4'. And just outside we have that 5' sugar. Over here will be my nitrogenous base. And over here will be my phosphate functional group. My next nucleotide monomer is going to come in directly above. Now in your notebook at home, I hope that you are trying to draw these upside down and not cheating by flipping your notebook over and drawing them in the opposite direction. This process of drawing upside down is actually a technique that people practice in professional art classes. What it does is it forces you to see and understand things in a different way. I have another cat knocking at the door. I'm going to let him in. Even building a DNA molecule is interesting to these animals who are joining us today. So in our second strand of DNA, we are still building it from the 5 carbon to the 3' carbon. But because it goes in the opposite direction, because this strand is anti-parallel to our first strand that we started with, this strand is harder to make, and it is called the lagging strand. And the other term that we see associated with this is the anti-parallel strand. So here we have one DNA molecule that we have built and we have made. The next step that we're going to cover in our next video will be how we make copies of DNA, how we do the DNA replication process. Again, if you have any questions, please email me. Watch this video again, look through the lecture slides, email me, let me know if you need any help.